So with your permission, for a few minutes I'm going to talk Turkish, and then I'll come back to the panel. Bugün benim için de çok önemli bir gün, değişik açılardan. Ben yaklaşık 40 yıldır Amerika'da yaşıyorum. İlaç şirketinde son 25 yıldır Pfizer'de çalışıyorum. Son 2 yıla kadar çalıştım, emekli oldum oradan. Bu süreçte bir sürü klinik araştırmaların parçası oldum. Ancak Türkiye'de ve Trustlife'da şimdi bu yolculuğa bu şekilde devam edip birinci faza girmek çok özel bir şey benim için. Herkes için bunun tabii farklı özellikleri var. Benim için Türkiye'ye sık geliyorum. Bu gelişlerdeki anlamı da artıran bir durum bu. Ülkemize hayırlı olsun. Destekleriniz için teşekkürler. Benim Trustlife'la ilişkim yaklaşık 6 yıl önce Adil Bey'le Harvard'da bir toplantıda tanışmamızla başlamıştı. Ve çok güzel bir beraberlik oldu. Ben de bu beraberlikte Trustlife'ın bir parçası olmaktan gerçekten gurur duyuyorum. Şimdi izninizle panele geri dönelim. Okay, so now what we're going to do is I'd like to introduce the speakers a little more broadly. I think it deserves a little more introduction. I also want to say this is an incredible panel. I'm so honored to be actually privileged to be moderating such a panel. I think we have collectively close to 200 years of experience on this panel right now. It's really tough to bring this group together. I'll start with Bernhard to my left. So Bernhard Palsson is a professor of bioengineering and an adjunct professor of medicine and a principal investigator of the Systems Biology Research Group at the University of California in San Diego. And so he and I share a passion for San Diego. That's where I live as well. He also serves as the CEO of the Novo Nordisk Center for Biosustainability in Denmark. He is the author of over 40 US patents and founder of several biotech companies. His research includes the development of methods to analyze metabolic dynamics and formulation of complete models of selected cells of several human pathogens. Dr. Palsson is a member of the National Academy of Engineering and is a fellow of several other organizations, including the American Association for the Advancement of Science. And to his left is Professor Leroy Hood. I studied his papers and read all his things and listened to him in a lot of talks. It's an honor to have him here, sit next to him here. He's a world-renowned scientist and a recipient of the National Medal of Science in the U.S. in 2011. That is the highest honor the U.S. bestows on a scientist by the President of the United States. He co-founded the Institute of Systems Biology in 2000, a member of the National Academy of Sciences, National Academy of Engineering, and National Academy of Medicine, which makes him, out of the about 6,000 scientists worldwide who belong to one or the other, he's actually in a group of about 20 people who's a member of each one of them. Dr. Hood is on faculty. He was on faculty at Caltech in California. He and his colleagues developed 4 sequencers and synthesizer instruments that paved the way for the Human Genome Project's successful mapping and understanding of the human genome. Professor Hood is currently carrying out studies in Alzheimer's disease, cancers, and wellness. He has played a role in founding 15 biotechnology companies, including Amgen, which I'm sure you all know, Applied Biosystems, which I also think you know, and NanoString. So welcome. And to his left is Professor Mattias Öhlund. He's at the KTH Royal Institute of Technology in Stockholm, Sweden, where he conducts basic and applied research in protein science, antibody engineering, and precision medicine. His innovations include antibody purification and next-generation sequencing, antibody-based proteomics, and AI-based drug development. He initiated and led the Human Protein Atlas, which is very important for Trustlife and the work that's been carried out since 2003. He's a co-founder of 21 biotech companies, member of the National Academy of Engineering in the USA and Royal Swedish Academy of Science, and the Swedish Academy of Engineering Science in the European Molecular Biology Organization. He was the founding director of the Science for Life Laboratory, a Swedish national infrastructure for molecular biomedicine, bioscience. And to his left, last but not least, is Cem Öztürk, currently the head of Sanofi Eurasia and oversees a multi-country organization encompassing Turkey, Russia, the Commonwealth of Independent States, and the Levant region, I'm told, about 90 countries. I don't know if he's been to all of them. He has held pivotal leadership roles in strategy, business development, finance, and sales and marketing over 20 years across Europe, Asia, Middle East, and Africa. He completed an MBA at Sorbonne University in France and started his career in banking approximately 25 years ago. He joined Sanofi in 2002, resides here in Istanbul. His role covers a vast portfolio including rare diseases, diabetes, cardiology, and strong emphasis on immunology, all guided by a strategic and agile management approach. His expertise now fuels initiatives that bolster social health and economic vitality while nurturing emerging talents. Again, I'm very happy to be sitting here with you, and thank you for making time to travel here today. So I'd like to start with Matthias and ask him, what role do publicly available data sets play in drug discovery? And in that context, perhaps you can describe the Human Protein Atlas and how it contributes to discovery of drug targets.
Okay, can you hear me? Yes. So it is, of course, really nice to be here. I just want to start by saying that I've been working with Turkish scientists for now almost 30 years, and I've always been so impressed about the devotion and the competence of Turkish scientists. So I am the director of an effort in Sweden called the Human Protein Atlas. It has been an effort, and we got a lot of funding, about $200 million from a non-profit organization in Sweden. It's the Wallenberg family. It's a very famous financial family. And they wanted to put money into trying to define the building blocks of human beings, the proteins. So all of you in this audience have about 20,000 building blocks that make up and makes you go and wake up in the morning and have coffee and so on. 20,000 building blocks. Almost all drugs in the world are directed to these proteins. You activate these proteins or you inactivate the proteins. That is how drug development is working. So what we have done in this Swedish project, but we have collaborated with Turkey, with US, a lot of collaborations with China, South Korea, and Japan, and so on, is to then try to map all the human building blocks, 20,000 proteins. We have created then an open access resource because we think this information should be available for everybody, all researchers in the world that wants to develop the next generation drugs. So we have now 5 million web pages available for all of you. You can go home tonight and you can go in and click around. It will take you some time to go through 5 million web pages, but you can of course go for your favorite protein if you want. So this is the background of the Protein Atlas, and of course this is a new era in biology and medicine where people, researchers, and research groups are creating a lot of data, and this is now available for everyone. You can actually access it and you can use it to develop the next generation drugs. And this is, I guess, what we will talk about today then, is how you do this, how you can actually use AI, machine learning, systems biology in order for a new era where we will actually define the next drugs in the future. So I don't know. I think I can start with that. And
thank you.
Yeah.
Thank you. So we heard the term systems biology quite a few times today. Maybe I'll ask Bernhard to tell us a little bit about what systems biology is and how it helps in our efforts for drug development.
Sure. Briefly, between 1950 and 2000, we had the golden era of molecular biology, where molecular components of cells were defined and characterized. Then around 2000, we got the whole genome sequences, so we could find the genes in a particular organism and therefore get an information about the collection of molecules in that organism, and we started realizing they all work together with one another, and we could reconstruct those interactions, and then we realized that these are all systems. So the cell is really a system with a bunch of subsystems in it. And since then, we have been developing experimental and computational tools to meet these challenges. As mentioned, we have a number of statistical approaches and first principles approaches to computation. We have an ever-growing availability of databases with high precision data in it. And now we stand at the cusp of deploying these tools to impact drug development, discovery and development. And if you want any examples of that, I'll be happy to mention one or two.
Absolutely.
So drugs have side effects, as we all know, and maybe one of the more famous side effects is what levodopa does to Parkinson's patients. They get this dyskinesia, as it's called, this shakiness. And this can be elucidated by using systems biology. So you take human neuronal cells, put them into culture, expose them to levodopa, and you compare the cells that have been exposed and not exposed. And by doing what's called expression profiling, that profiles all the genes activated under that condition, you can actually find out what levodopa does to the cells. And when you look at that, you can find out which processes are activated and related to dyskinesia. Then you can go in and screen thousands of other molecules to see if you can find one that suppresses what levodopa upregulates. And that takes some work, but you can find such molecules, and we have. And that is now about to go into investigational new drug approval at the NIH. And this kind of discovery would help create an adjuvant drug that will be taken with levodopa to treat Parkinson's disease. So that's one example of a kind of high precision systems approach to drug discovery and development.
Thank you. Lee, I'd like to come to you with a question. So would you take us through how you use the Human Metabolic Atlas and the systems biology to integrate and interpret big data to arrive at drug targets? And of course, technologies related to that. You can also tell us a little bit about.
Well, I think this is a real tipping point in biology where we have systems approaches put together with AI, put together with striking new powerful technologies that look at very large dimensions of data sets, and of course, the ability to focus these things on wellness or prevention or disease. I started a nonprofit about 4 years ago to do data-driven health. And the idea here is we take the genome and phenome from each individual, the genome defining the potential of your health trajectory, and then taking you from birth to death and identifying just where you are on that trajectory with the phenome. And the phenome is governed by two major factors, your behavior and your environment. And we can measure blood analytes, we can measure the gut microbiome, and we can measure digital health for both brain and body. We used systems biology then to take this enormous mass of data and map it into the different biological networks, each of which is one-dimensional. There are 4 major types, and the biological communities that bring together all the different dimensions of data features. And these gave us fundamental new insights. One, into the actionable possibilities you can use to improve human health. And two, hundreds, and now we're getting to thousands of potential drug targets that are focused for me on wellness and on prevention. With a company I started called Airavail, 5,000 people we analyzed over a 4-year period, we came to 3 major conclusions. One, the genome has tens of thousands of these actionable possibilities and new drug targets. Number 2, we were able to use the mapping of the networks and the communities to be able, again, to identify hundreds of actionable possibilities and targets, again, focused on wellness. And the third really striking observation was we could do the very earliest detection in the transition from wellness to all major chronic diseases by 3 different techniques, which gave us insights into the original networks perturbed, so you could focus on preventive therapies that reversed them before they ever clinically manifest. And this led to the idea that we'll replace our current disease focus with one of wellness and prevention. Now the question is, with all of the drug targets we've generated, where we're going. So a second dimension was creating a company called P4 Bios, which actually uses peptide drugs to really, I think, transform how we're going about in the pharmaceutical industry in the future. We're looking at millions, if not billions, of 30-mer peptides whose unique feature is they're unstructured, and they have a shape-shifting ability to interact with the 60% of the proteome that is actually unstructured itself. And as well, they can interact with nucleic acids of many different types, the RNAs and so forth, and all of the proteins and the 14,000 mini proteins that have been discovered. And the idea is to take a technology we developed in the early 2000s using inkjet technology to be able to synthesize 1 million or 10 million DNA fragments on a chip, genes for the 30-mer peptides. And we can take these individual genes and place them in a lentivirus. We can infect 1 million human cells with 1 million different drugs. And we can then do phenotypic selection. For example, we can see which of the drugs transfer a cancer cell to become a completely normal cell. Once we have those cells that have selected in a single shot from millions or tens of millions of different drugs, the 1 or 2 or 3 that are optimal, we have enormous mutational space dimension for optimizing that drug away from side effects to focus enormously just on the target you'd like it to focus on. And we have finally the ability to take each of 10 million of these different drugs and place them individually in 100 different human cell lines, gain their transcriptional patterns, feed this set of transcriptional patterns into knowledge graphs that connect these transcriptions with, again, the networks and the communities, and then use that to instruct transformers to get agents then that can, 1, predict, given a peptide, what its transcription factor features are, and 2, given from phenome health, certain transcription patterns we'd like to mimic, what peptides we have to use for it. And of course, what this does in a very powerful way then is gives us, with Transformer, the ability to look at the genome and phenome of an individual and prioritize from the millions of different actionable possibilities those relevant to the individual, while at the same time we can do exactly the same with the peptide drugs, determining which that individual needs in the context of their genome and phenome. Thus, we tie together the genome, the phenome, and AI and systems biology with powerful new approaches to drug targets. What I'm excited about is the possibility we could really collaborate with the company that we're seeing here in the future to realize some of the powerful advantages as we move forward.
Thank you. So there's been a lot of change in technology. In my, in my short career of about 30 years, I've seen technology move from looking at microsatellites to SNPs to being able to completely sequence the human genome. All the other technologies that have become available are generating an enormous amount of data. You can generate on a person, just one person, millions of data points, if not actually nearing billions. So with all this and the technologies and the systems biology we talked about, as well as big data, Jem, I'd like you to tell us a little bit about how these new technologies and the availability of big data is changing how we discover and develop drugs in the modern day pharma, perhaps with specific reference to Sanofi, if you like.
Yeah, thank you very much. Before answering to your question, I would like to say that I'm super proud of being here. I have participated to many panels, but that one is a little bit special to me because I remember the days that Adil came to see me 4 years ago, right, Adil? Talking about his dream. And to see the evolution and to see such an audience and such valuable guests, partners, investors around the table, for me, it's a dream that he was having that has been realized. I would like just to give him and to hold him a good round of applause. And thank you very much for having us here. So coming back to what's happening with the technology and the way that it's evolving. To be honest, we are a bit scared about the speed that it's evolving. Because Bill Gates was saying that the technology, the speed that it has been evolving over the last 100 years, what we have seen over the last 100 years, we will see it happening in the next 10 years, in fact. So to cope with that speed as a company alone, it's almost impossible. So what we are seeing more and more in the industry, Sanofi is a company that has been growing as maybe for some of the people who doesn't know very well Sanofi is a #1 French pharmaceutical company, and it's in the top 10 in the world, generating 44 billion euro sales and having from rare disease to immunology, cardiology, diabetes, as you said, Hakan, operating in many disease areas. But Sanofi has been growing over the last 40, 50 years with mergers and acquisition, buying some different pharmaceutical companies. But over the last years, what we are seeing is what we are doing, we are partnering with a lot of AI companies. We are partnering with a lot of startups because we understood very well that doing everything alone with your own data and with your own scientists, it's almost impossible. And the good example of that was COVID. During the COVID process, everybody was expecting the COVID cure coming from the type of big companies, what we call the Phoenix ones. But for a Phoenix company to take a decision and to do it in an agile way, it's not always easy. The solution came from startups, what we call unicorn companies. That's why, in fact, we have started to see more and more collaboration across the industry, and we have started to see more and more acquisition and more innovative ideas that have been adopted in cooperation with big companies collaborating with small companies. What we have done very recently, in fact, we have invested in many different areas. For example, one of them is Amnix. They are using a physical algorithms to discover small molecules, and same another deal we have done with Exscientia, and more or less we invested close to $1 billion to those investments. But what I'm proud of to see is, and it's a concrete result, almost 70% of the new small molecules that Sanofi is running after and is discovering is coming from AI discovery. That's why what I'm seeing here is giving me a lot of hopes. It's true that we are developing something here internally, and we are trying to create our future as a country to launch a molecule in Turkey, to develop a molecule in Turkey, and afterwards to export probably this molecule outside of the country. But on the other hand, we are also targeting some unmet needs. And that is an area that is pleasing me very much. We have great scientists here, but to target those areas, prostate cancer, Alzheimer, muscle sarcopenia, etc., is not easy. I'm sure that there are some people around you that are having this kind of diseases in their family, in their friends, in their close loop people. My mother is at the hospital now, and she's suffering from muscle sarcopenia. And this is an area that we don't have a cure. And as companies, nobody is investing in that area because it's very costly. The cost of a medicine today is changing between 2 billion to 20 billion euro. And we have Selahattin here that I know very well as an investor. And when you look to the investment, sometimes you invest 20 billion euro and you get nothing. Because in the phase 3, you get a side effect that make you drop everything. That's why the usage of big data, the way that we are analyzing, and even the new technology is super important because it's reducing the probability of failure to you. It's reducing the time, it's reducing the cost, and the probability of failure. And you choose the right area to invest. That's why we continue to cooperate with innovative new startups. And I hope that, as you told, I'm the head of IFAD in Turkey. All the big corporations are looking also for new innovation. And those kind of startups like Trustlife is very, very important for each and every one, because a small discovery here with the help and collaboration of a big company can be a big discovery for the humanity in the future.
Thank you. I think, Matthias, you want to say something?
Since we're talking about technology, I just want to make a comment here. So I think we're all amazed about what is happening in AI and the large language models and ChatGPT and so on. The speed of technology development is just staggering, and it is just so impressive, almost scary. But I don't think that most people realize that we have almost the same or even higher speed in life science and medical technology. So when Leroy Hood, I was also involved when we did the first genome project. It costed us the society $3 billion, an enormous amount of money. Here we are just a few years later, and each one of us in this room can do a whole genome for $1,000. It's a factor of 1 million cheaper. It is just amazing. But what I think even less people know is that this is on the DNA. And DNA is interesting. This is what we got from the father and mother. We got half of it. And that is why we are who we are, and so on and so on. But of course, from a medical point of view, it's the building blocks of life that are very interesting, and that's the proteins. I'm a protein guy, so I'm a little bit biased here. And here we've been lacking technology. And to say we want to analyze many proteins simultaneously, and this technology has simply been lacking. And to give you a flavor of that is that there was a very famous company in California, where these guys come from, called Theranos. 10 years ago, they claimed that they could, from a little blood, they could actually analyze 100 proteins. That was amazing. They started a company. The company became $10 billion value, but it was not possible to do it. It was a bluff. And the founder of that company ended up in jail. Here we are, 10 years later, and we can do 11,000 proteins in a small drop of blood. That is a factor of 1,000. And this is a reality now. So it is changing like crazy. And that, of course, opens up a lot of new developments and also new technologies and new adventures. And this is the sort of adventures that Trustlife is part of, that the ones we in the panel are part of. And it is so nice that Turkey is part of this journey, and you're taking part in the future of developing the pharmaceuticals for the future. It is very exciting.
Okay.
Yes, of course.
So I think one of the enormously important areas of technology development is the development of wearables. We recently received from ARPA-H a $55 million grant, and one of the projects is to take the insights about the pancreas and the activity of the beta cell that we can infer from this massively wide multi-omic analysis, and take wearables that have all sorts of signals. No one has any idea what they're about, and do an impedance match so you can use the wearables to be able to gain that kind of insight mediated by a cell phone. And we can do that for the brain, we can do it for the body, we can do it for the gut microbiome, the 3 areas of health. And in doing so, it democratizes data-driven health in the sense you can think about taking it to underdeveloped countries that have cell phones for most of their citizens, and you don't have to take them in and analyze complicated blood and things. So I think the wearables, there's one wearable coming out that can do blood pressure now absolutely beautifully. So you can get not only that data transfer, but you can get continuous monitoring that take into account the very short-term rhythms that exist in a human being, as well as longer-term rhythms.
So it's pretty clear that availability of big data and technology is changing the way drug discovery and development is carried out these days. So the traditional drug discovery and development involves a lot of chemistry. And that's been the basis of how pharmaceuticals have been developed so far. Now we have a new world where we're dealing with a lot of big data and they have the opportunity to start with big data first, identify targets, then come back and do the chemistry necessary to that. So I wonder if, Bernhard, you could comment on how this, the big data, systems biology, use of other technologies is reshaping the way we're actually discovering medicines, and also how that may help us in reducing toxicity as we do so.
Okay, so let me maybe add another group of diseases that has been kind of ignored until COVID, and that's infectious disease that is relatively unsophisticated as a field compared to, say, cancer or heart disease and so forth. But now we know that antimicrobial resistance is likely to be a defining challenge for medicine in the 21st century. So we need to develop new medicines there, develop databases, and build them out. So in the United States, we handle about 2 million bacterial infections a year, and there's no reason why we wouldn't sequence all these bacteria. And on a global basis, it's maybe 50 to 100 million. So we can very quickly generate the genetic basis for how infectious bacteria work. And if you can couple that now to new data types like treatments and clinical outcomes, you can probably apply machine learning and AI to almost personalize the treatment for a particular pathogen and the way it's treated. Now we know that all bacteria will respond to antibiotics differently, so we can also use a similar approach as I mentioned earlier to understanding the systems in bacteria that respond to antibiotics. And we do that by simply exposing them to antibiotics and looking at the gene expression. So I think there is going to be an enormous impact of big data analytics on the understanding and the treatment of infectious disease, going all the way from understanding how the individual bacteria respond to drugs, and if you identify it in a person, you will have maybe many clinical histories already on a database that would show treatment modalities and outcomes, or how that particular bacteria was treated. And with antibiotics, it isn't maybe so much new drugs that you need, but it's more proper use of them, called antibiotic stewardship. So we could see big data just cast a net across the globe and fully expose how these bacteria are moving around, how they infect people, and what is the best way to treat them on a personal basis.
So I'd like to shift gears a little bit and talk about implications of big data. So we obviously get a lot of data from various organisms. We also get data from people. So many years ago, I did my 23andMe genetic test. To my surprise, I found about all my genetic background, which I thought was Turkish, but there was nothing listed there for that. But that's a different topic. Recently, the company got sold, so it was insolvent, and now it's in the process of being sold. So people are advised to block and have their data destroyed, because you have all your genetic data available to whoever buys the company. They can use the data in whatever way they like to. So actually, I did take all my data out, or at least requested that to be done. So there is that data privacy, data security issues. This affects all of us, anybody who's involved in a trial. And with Trustlife in Turkey going forward with clinical trials, I believe this will be also top of mind for a lot of people participating in clinical trials. So I'd like to pose a question to Jem as representing here, representative of a big pharmaceutical company. How do you deal with security and data privacy issues, and how do you assure clinical trial participants that their data will be safe?
Thanks, Hakan. I believe it's first it's a very good and also a difficult question, because it's an area that sometimes we tend to see as a bureaucracy. But in reality, if you take it in all aspects, it's a strategic leverage. Because at the end of the day, if you are not respecting this kind of international standards in a country or in a company, you put yourself at risk, but you put also yourself in a position that you cannot be, in fact, an international player if you do not respect this kind of rules. We are operating in an industry that's requires a lot of responsibilities. And in terms of this responsibility, if we are not obeying to this kind of rules, at the end of the day, you lose trust. I think you gave a very good example, right? A company that started and they said that they can analyze the protein and analyze that kind of data, etc., reached 10 billion. At the end of the day, the guy ended in jail. That's why it's very important to get the consent of the people into the sake of transparency and to be publicly opened everywhere in a transparent way. Otherwise, I mean, you lose trust of the people. What is very interesting in clinical trials, etc., and we made a research in FBA as a global analysis, and we asked the people, would you like to be part of the clinical trials, knowing that this data will be used for the good of the people? 65% of the people, they answer, if really it's for the sake of humanity and they believe into the project, they are part of it. So our duty is to convince the people that there is a good purpose of it in a transparent way, and then getting their consent and to put publicly this transparency to everyone and to obey the international rules and to be an international player in whatever we do.
Matthias, you wanted to say something?
Yeah, just to comment on that, because it's a very, very important question, actually. And one of the problems we have with the new technology, we will actually know so much about the people that we are analyzing. And that is an ethical problem. And in the ethical committees, at least in the north of Europe that I know, are very careful that the information about a person will not go back to the person, so it's anonymous. But it's, of course, with all this data, it is still very hard to protect that you don't know exactly who you are actually from the data. So we spend a lot of time with the ethical committees to actually make sure that when we analyze that the data which is open access, the person who has been investigated cannot actually know who he or she is. And this is, but I think you can do it, and there's good ways to do it. And obviously for mankind, this is very important. We can actually analyze and couple metadata, which is the data about the person, with the genome data and the proteome data. But we do spend a lot of time with this actually right now. But it is solvable in different ways. And especially for us in the Protein Atlas, we have 5 million web pages. And of course, a lot of data about patients and individuals, and it's open access. Everyone in this room can go in and look for 5 million web pages tonight, if you want. But of course, what we really have to take care of is that from that data, you cannot find yourself in that data. So this is something, but we can deal with it. It is a lot of bureaucracy, but I think it's good bureaucracy. It is to protect the patient from itself.
Yeah.
There is also a learning curve, and it reminds me a case that was in 2021. One of the big pharma and in oncology, I mean, they did a clinical study and they used full anonymous data with some screenshots of the patient results. But the AI is evolving, and with the AI, with an algorithm, they were able to find which hospital, which region, which type of genetic patient, which origin, etc., and almost to be able to find the patient. That's why the authorities entered into the game, and the clinical studies has been delayed by 3 years almost. Yeah. So sometimes, as you say, it's a bureaucracy, but it's a good bureaucracy that should be respected, but there is also a learning curve.
But it is interesting because in the project, the precision medicine project that we're running, we have analyzed 20,000 patients with 70 different diseases. It took us about 2.5 years to go through the bureaucracy to do it. Then it took a few months to do the analysis, and now we're on the 2nd year of analyzing the data. So it shows a little bit that this has completely changed the world for us being biological researchers. So much is on the ethics. So little, actually, with the new instruments is on the actual analysis, which used to be the 90%, and then so much is on the downstream. But on the downstream, of course, we now have new tools, AI, machine learning, and so on.
Yes, Lee?
So let me make a point about big data that I think is really important. When you create a set of big data for a series of individuals, it is enormously valuable. The 5,000 patients we looked at for 4 years, we've had more than 30 peer-reviewed publications come out of that. And others have used that data in exactly the same way. And I mean, you can look at so many different ways at a given data set. So making the data public should be central to everything we do. For-profits, of course, have to skim off the low-hanging fruit, but it's absolutely essential these big data sets become generally available, because it's thousands of people looking at that get, I mean, human biology is so unbelievably complicated. One of the big debates in AI now that I have with Craig Mundy, who was #2 at Microsoft and started OpenAI, is whether or not the AI factors, the transformers, will get to be 10 times smarter in the sense of being able to integrate data than we can within the next year or two. And I think the answer is they will. And he's arguing now that ultimately AI will be able to generate causal relationships without any biological experiments. And I think it's utter nonsense because of the incredible complexity of human biology. Because remember, evolution is opportunistic. It builds on what's there. It doesn't care about the law of Occam's razor. It puts together regulatory systems that are unbelievably complex because that was the easiest way to get there. And in order to decipher all that complexity, we have to generate entirely new windows into the data that we don't have yet. And I'll guarantee, no matter how smart the AI machines are, they'll never solve that problem of human complexity without enormously more and different types of data.
Right. Bernhard? So maybe I'll make an observation here, since you were talking about the cultural changes and what's ahead. So let's think about human resources and training a little bit. So if you're generating data, the effort is linear with the number of data sets you want. If you want 10, it takes so many hours. If you want 20, it's twice. So they scale linearly with size. Might even bend a little bit down because you get better and better at it. But imagine if you have 100 transcriptomic data sets and 100 proteomic data sets, you just don't compare the conditions, you have to compare the whole data set. So now you have n squared as the analysis problem. Then if you add a third data set, you still have 3 times n in generating the data, but now you have n to the third power in terms of the analysis. So when you model out the workflows associated with true multi-omic data and large data sets, before you know it, the data analysts need 95% of the resource or even more. And especially as data is getting socialized through public repositories, it's easier and easier to get the data, but it's more and more challenging to analyze it. And I think we all have also, with respect to analysis, we all have the same caution as Lee just brought up now. Many of these data sets are not nearly large enough to do transformer models. So chat, for instance, has all the issues of the New York Times going back forever, all of the Shakespeare writings, everything. And we may not have nearly the same size of data or amount of data to be able to do good transformers. So knowledge enrichment of the data based on first principles and what we know about the molecules and so forth will be very important. And as of now, it's not obvious how you systematize that. So the manual curation of the data you were talking about is still going to be a factor. And that's going to even increase the need for data scientists for data analysis and interpretation. I just want to mention this because this is routinely overlooked.
Yes.
Just to comment on that. So 15 years ago, I approached the government of Sweden and said that the future of life science is data-driven. We need to focus on data-driven. And the government of Sweden decided then to support a new infrastructure in Sweden called the Science for Life Laboratory. I was the employee number 1. And then we tried then. Now we are only at the Karolinska site at the SciLife Lab. We are 1,700 people. So this just shows you how important it is to be able to do data-driven life science. And one of the most important things is to get young scientists that knows about AI and so on. So for every group leader position we have at SciLife Lab, we have more than 100 applicants. It's more than 80 different countries that the group leaders come from. And one of the most talented of the group leaders at SciLife Lab comes from Turkey. And he's in this room. It's Adel Madinoglu. So.
So in the last few minutes, I'd like to maybe look into the future a little bit. And I'm a little impatient with future. I don't want to look 20 years from now, 10 years from now. I'd like you to think about the next 3 to 5 years. And given the enormous changes in data generation and the use of data in our current work, what do you see coming next? So we have, everyone talks about AI, everyone talks about machine learning, everyone talks about big data, and big data, it comes in the form of multiomics, and that's a lot of different technologies. So where do you see it going? And if where it's going versus where you like it to go are different, where would you like it to go? I think I'll go just down the line here, starting with Bernhard. So look 3 to 5 years from now.
Okay, so we have data and we have analysis methods. On the data side, standardization, reproducibility of data from site to site and so forth will continue to be a problem. There are some exceptions from this, like sequencing data, whether it's RNA or DNA, is easy to standardize. There are some phenotypic data that can be made reproducible from lab to lab, but the quality of the data and the access to it. On the algorithm side, there will be, I think, the dynamics will be very interesting to see what's going to happen, and it's actually going to be hard to predict. I mean, just 3 years ago, none of us were thinking about transformers and AI and so forth. But I think, so I don't really want to forecast that other than we're going to see a lot of interesting changes there, which is a combination of first principles and statistics. I will just maybe make one observation, and that is that unsupervised AI will hallucinate, as we know. It will generate things that aren't right, and we see that often, of course, when we have chat write text for us. So I think it's very important to get first principles constraints on data sets so we don't start regressing outside of physical reality.
And Lee?
So I would say two things. I think within the next 10 years, we'll see a healthcare that's more and more focused on wellness and prevention. And this is going to answer, it seems to me, the 5 major issues that face health in society today. One is the quality of healthcare. It'll be increased enormously. The second is our aging populations. And we already see that this scientific wellness I'm talking about is the other dimension of healthy aging. The third is the explosion of chronic diseases. In 10 years, I can imagine 50% of the chronic diseases have been avoided by early detection. And finally, the whole question of cost. If you can avoid chronic diseases, you can save trillions of dollars in the future. And this can be invested in all of the things that will optimize a healthcare of wellness and prevention. And on the other side, I see this peptide drug approach that I've talked about here that has scale, that has the ability to do one-shot selections of billions of peptides, that has the ability to optimize the peptides to avoid side effects. And I think there are 3 ways we can do that. We can talk about that sometime. And more than that, to feed what the drugs do in the human body into a transformer together after connecting through knowledge graphs, them with the entire knowledge of networks and communities and so forth, we'll have the ability to apply that to the individual too, in a unique, powerful ability to pick drugs in the context of what you are with regard to genome and phenome, and that creates a N of 1 healthcare that is true individual healthcare that I think is going to change the world, and I think we'll be able to democratize this and move it from the developed to the undeveloped countries as well, if we don't get too many dictators in the system.
Matthias?
So when I was on the board of directors for a company called Pharmacia about 25 years ago, and in the board we discussed a lot the future of drug development and so on. And the world that we talked about was actually something coming from Leroy Hood here, precision medicine. And what we said at the time, that we have to transform the medical industry to actually make the physician to actually make wiser decisions. Who should get what drug and so on. I would say that a lot of things have happened in 25 years, but we haven't come really to what I think is the sort of the end of the road of precision medicine, where you do early diagnosis of a disease even before symptoms, and you stratify patients to tell this should have this drug and this should have this drug. I know it has been rather slow for 25 years, but what is going on now with the new technologies, all the things that we do, the open science and everything, I think we will discover, or we will see, a fantastic development in pharmaceutical industry in the next 10 years. And I think Turkey should be in that journey.
Thank you. And Jem?
I cannot agree more. I believe that there will be some fantastic development. I like the way that you ask where would you like it to go and what do you think it will happen. So it's two different ways, right? And sometimes some people are saying to us when we are in this kind of panel, 5 years later there will be a technology that I can be healthier and I can look younger. So these are the two things that we are running after, right? In reality, we are running after immortality, but that one we gave up. So we want to be living longer and healthier. So what I believe that in the near future, with the way that the technology is advancing, we will find a lot of good solutions in terms of healthcare. And maybe some of the diseases that we are talking today as mortal, incurable, will be curable. There will be some new ones. But unfortunately, on the other hand, what I was thinking is that Bülent has shown very well with numbers the way that the world will be evolving, increasing population, elderly population, but increasing also inequalities. As of today, unfortunately, the life expectancy in the developed country compared to undeveloped country is very, very big. With my experience, I have been all across the world, and I have been also leading in my past Africa. And Nigeria, which is one of the key countries over there, life expectancy is still 55 years. When you go to Japan, you go to Singapore, etc., you are talking about 86 years. So we have a good way of saying, where are you from in Turkey? This is the first thing that people ask you. And where are you from? Which city, exactly which area? So we say memleket nere in Turkish. So this memleket nere, where are you from part, is the place that you are born is changing your life expectancy almost 1.5 times more. If you are born in Japan, you live 1.5 times more if you are born in Nigeria. So I hope that pharma industry will contribute in a way that sharing the data, between ourselves, because as multinational companies, we are all having data. Startups are all having data, but everybody is trying to have their own data. And they are not sharing we are not sharing between ourselves. If we are more sharing and if we are more working together collaboratively, as we have been doing during this COVID period of time against the pandemic, I think we can be much more efficient and solve this inequality of the world and increase our life expectancy and to live in a much better world.
Yeah. Well, we got 20 seconds left. So first off, this has been an absolute pleasure and a privilege to have you all on this panel. And thank you for the opportunity for us to have this discussion. So I'd like to thank Bernhard and Lee and Matthias and Jem. And I hope you join me thanking the panel for this great panel.